fight against plant diseases doesn't rely heavily on harmful chemicals, but tries its best to harness the power of nature itself. Ladies and gentlemen, a pleasant morning to you all. I am Kadeen Jardine and this is my colleague Helena Kunga. Today we'll be presenting our graduation project entitled In Vitro Effects of a Bio-Input Enriched with Beneficial Microorganisms on Pseudocercospora fijiensis in bananas and Colletotrichum coccodes in tomatoes.
And here's the outline of our presentation. We'll start with an introduction, go to objectives, the methodology, results and discussion, finally a conclusion and recommendations.
Now, I am sure that the majority of the persons in this room can attest to the fact that bananas play a key role in many countries, including those of the Latin American and the Caribbean region. This can be shown through the fact that in 2022, they had an exportation of over 15,000 tons, leading to great development in their country, whether it be in the area of food security or the generation of foreign exchange. However, in any production system, there will be challenges. When I speak of challenges, I am talking about diseases, such as Fusarium wilt, Mocha disease, and black secatoka. Throughout the duration of this presentation, we'll be focusing on black secatoka, also known as Pseudocercospora fijiensis, characterized by chlorotic specks after 14 to 20 days of infection, but leads to great negative effects in the plantation, whether it be in early maturity or the reduction in yields.
Well, similar to bananas, tomatoes are also one of the most consumed fruits in most parts of the world, including the LAC region. And according to Lopez Marin 2012, there was an annual production of 58 billion tons of tomatoes and a per capita consumption of 20 kgs. And this contributed towards the food security systems in these countries, as well as employment in both direct and indirect. As my colleague earlier stated, tomatoes are also affected by diseases such as early leaf blight, Fusarium wilt, and anthracnose, which is scientifically known as Colletotrichum coccodes. And this is known to be a cell-borne disease that is characterized by foliar necrosis and chlorosis in both leaves as well as fruits. And it is important to note that this disease results into yield reduction. And the fungus Colletotrichum is also known for its dormancy, that it appears later in fruits, affecting the post-harvest quality.
Now let's take a look at the strategies that persons have employed over the years to combat these diseases. We have them grouped into three main categories. We have cultural, chemical, and biological. In the case of cultural for black secatoka, we can see that persons try their best to have fertile soils and also to remove the affected leaves. Now when it comes down to anthracnose, as my colleague already mentioned, this is a soil-borne disease, so persons try their best to have soils with proper drainage and also to save the tomato crops. When we look at chemical, persons perceive the chemical strategy to be the most effective. And this is why in the LAC regions, they apply these chemicals 35 to 45 times annually. But this leads to great phytotoxicity and fungicide resistance.
Well, in recent agriculture systems, most farmers and researchers have tried to reduce the use of synthetic fungicide through the implementation of biological control strategies, such as the use of beneficial microorganisms, the use of bio-inputs, as well as plant extracts.
This therefore takes us directly into our objective. The general objective was to evaluate the effects of a bio-input enriched with beneficial microorganisms on Pseudocercospora fijiensis in bananas and Colletotrichum coccodes in tomatoes.
With specific objectives which included to isolate the beneficial microorganisms collected from two undisturbed ecosystems and then develop a bio-input that is enriched with beneficial microorganisms for controlling the two phytopathogens, and finally evaluate the effects of the bio-input on the growth and germination of the two phytopathogens.
Now it's time to tell you where exactly we did our investigation. We did it here at Enriched University, which has the following or these climatic conditions. However, we were working in two main areas, the natural science laboratory and also the water and soil laboratory.
And now let's take a look at the experimental design. We were working on two crops, which are bananas and tomatoes, and focused on two diseases, which are anthracnose and black secatoka. And we did our experiment in vitro and conducted two trials, which comprised of 8 treatments and 10 replicates.
It is now time for us to take you on a step-by-step journey of our methodology. We'll start off with the field experiment. Under the field experiment, we have the collection of microorganisms. These microorganisms were collected using the rice trap method, in which they were installed in undisturbed ecosystem and collected after 2 weeks. We went ahead to also collect the infected leaves. These were collected at the integrated organic farm, taking into consideration the characteristics of the phytopathogens which were described in the introduction.
And now let's go to the laboratory experiment. We started with the isolation of the microorganisms, and this was done based on the colors and did an identification using a laser microscope. We then isolated the phytopathogens, and this was done in potato dextrose agar, PDA, and then later we did incubate them. We then did the extraction of microorganisms, this using the stirring rod and water, and then we did the quantification using a Newber chamber and calculated the concentration. We then re-isolated the phytopathogens from the previously isolated samples, and this was also done on PDA and then later incubated.
Now we move on to the preparation of the bio-input. As you can see, we have a series of processes which started with the disinfection and the weighing of the botanical plants. Then we followed the procedures to later leave the bio-input to ferment for 7 days under aerobic condition. It is important that you take note of the materials that we use and the quantities at which we use them. We later did the preparation of the treatments, which started off with the preparation of the agar, also known as a medium. We followed all the procedures to later leave it at incubation at 25 degrees Celsius.
And as in regards to the data collection, we collected our data after one control had reached the borders, and a reminder that we did 2 trials, of which in both trials, black sigatoka reached the borders at 7 days, while anthracnose in the first trial after 25 days, and the second trial after 10 days.
And for snapshots of our statistical analysis, we used RStudio. We ran 6 models, but we selected the log normal error distribution due to the fact that this one had the lowest AICC. We then went ahead to back transform our data on with using a 95% confidence level, and later performed a Tukey test with a p-value of 0.05 to understand the differences that existed within the of the treatments within the data set.
Well, now let's go straight to one of the most interesting and important parts as it pertains to our project, that's the results and the discussion. To begin with, on the collection of beneficial microorganisms, we noticed that they had different colors present in the rice traps, and this indicated that there was a diverse number of microorganisms present. We then did an identification to know what species were present and found out that there were fungal species, bacteria species, as well as nematodes. We then took a step to identify some of the fungal species that were present and found out that there was Trichoderma sp as well as Penicillium sp.
So both of the microorganisms that were identified are said to have antifungal properties. Now, according to Bailey 2008 and other authors, they said that Trichoderma sp has the capacity to parasitize, conduct antibiosis, and also to suppress the growth of phytopathogenic fungi. When we look at Penicillium sp, it also has the capacity to create bioactive secondary metabolites. When we combine the characteristics of both of these microorganisms, we can see that they're an excellent choice for biological control.
And as in regards to the bio-input, we found out that it had a brown color, as you can see on the image on the screen, and also had a good smell, which indicated that it had undergone the perfect aerobic fermentation process. And for both trials, these characteristics were comparable.
And to understand the distribution that we had in our data set, we created boiling plots. On this boiling plot, you can see that there's a little bit different in both trials. However, in both trials, we can see that they had asymmetrical distribution. In the case for P. fijiensis versus the treatment.
And similar to coccodes, there was an asymmetrical distribution. The only differences we could see are the sizes of the violins, which indicated that for the first trial, the data set was widely spread as compared to the second trial.
And now let's continue with the results that we got from the statistical analysis. Let's start with P. fijiensis versus the treatment. We can see that for the treatment of the fortified bio-input, that it was statistically different from the control. Similar to that of the T-Rex Gold, which was also statistically different from the control. Now let's pay key attention to the treatment, which had the combination of the T-Rex Gold with the fortified bio-input. Here we can see that it was not statistically different from T-Rex Gold or the fortified bio-input as standalone treatment, but it was statistically different from the control.
And now let's take a look at the growth of C. coccodes versus the treatment. Firstly, we can see that the fortified bio-input had statistical differences compared to the control. We can also see that the T-Rex Gold had statistical differences compared to the fortified bio-input as well as the control. And for the combination of the T-Rex Gold and the fortified bio-input, it did not have any significant differences compared to the T-Rex Gold, but did have differences compared to the fortified bio-input as well as the control. Let's pay key attention to the 2 phytopathogens. For P. fijiensis, the fortified bio-input as a standalone worked better, while for C. coccodes, the T-Rex Gold as standalone worked better.
So what would have caused the differences that we saw in terms of the phytopathogens reacting to our bio-input? It's important to note that both of these phytopathogens are from the same taxonomic classification known as Ascomycota, but they differ when it comes down to their class. Now, according to Izaza 2016, when phytopathogens have different classes, this therefore means that they will have their unique or independent physiological and biochemical attributes, which allow them to have different reaction to antifungal properties. In this case, we're talking about our fortified bio-input. input.
We're reminded that despite the differences in the reactions, but the 45 input still worked at inhibiting the 2 phytopathogens. That could be attributable to its components. To begin with, the beneficial microorganisms. According to Samuelian 2016, he states that Trichoderma sp has been used to controlling different kinds of diseases, including Mycosphaera musicola, which falls under the similar class as that of P. fijiensis. Liang et al. In 2021, on the other hand, confirms that Penicillium sp has also been used for biocontrol for diseases such as Pellicularia oryzae, which falls under the similar class as that of C. coccodes. And now let's go to the next component, which is ginger. Ginger contains antifungal compounds known as gingerals, and according to Nishidono et al. 2018, the 8 gingerals and the 10 gingerals are the ones that have been so far proven to directly inhibit the growth and multiplication of these phytopathogens. And to confirm that, CEFU in 2015 conducted a study using ginger oil at a concentration of 0.45%, and this resulted into a decrease in disease incidence to 38.1%.
Now, similar to gingers, aloe vera also have antifungal properties known as aloeamodine and aloe A. These are said to have antifungal properties, and many studies have been investigating these compounds to find out what exactly they can do. So let's see 2 examples that we brought today. So they use aloe vera extracts at a concentration of 5,000 ppm, where they were able to reduce the growth of Colletotrichum species by 7.2%. Also, in another study done by Jamarillo et al. In 2017, they found out that when they used aloe vera extract at concentration ranging from 500 to 1,000 ppm, they were able to reduce the growth of M. fijiensis, which is similar to P. fijiensis, from 30%, inhibition rates ranging from 30 to 44%.
And now let's go to the last component, which are cloves. Cloves contain antifungal compounds known as eugenils, and different studies have been used to prove the use of clove oil. And in a study conducted by Diora in 2009, there was an inhibition rate of 92.67% on M. fijiensis, and another study confirmed by Davy et al. In 2020 that there was a 100% inhibition rate for the Colletotrichum species.
It is important that we say to you that the 3 botanical plants that we use have similar modes of action in terms of they react with the lipids within the cell membrane of the phytopathogens and cause cell alteration of the cell structure of the phytopathogen, which therefore leads to cell lysis. So today we gave you a brief introduction, we gave you methodology, results, and discussion. Now it's time for us to leave with you conclusion and some recommendation. To start with that aspect, it's important that we state that all the treatments within our investigation had inhibitory effects against both phytopathogens. But when we looked closer, we saw that in case of the fortified by input, it had a greater efficacy on P. fijiensis as compared to C. coccodes.
We also saw that the combination of the 45 bio input with the Timoric goat had promising results as it pertains to the inhibition rates of the 2 phytopathogens. And we also noticed that the use of rice traps as a method for collecting microorganisms is very effective and efficient.
And for the recommendation, firstly, we must say to the person that will continue investigations like these or this same investigation that they try their best to use a wide variety or a wide range of concentration rates, as this will help them to understand or to know at which concentration rate they can get the highest efficacy.
We also recommend that this experiment be replicated in vitro in order for us to be able to validate the results that we got. And we also recommend that it also be done in vivo conditions to see if this particular bioinput would work in such conditions.
Ladies and gentlemen, today my colleague and I remind you of the importance of bioinputs and plant extracts. It is now time for us to take investigations like these to another level, to fight, to combat plant diseases with the power of nature itself.
Thank you so much for your